Okay, so we are ready for the second session where we will start looking at the future and how we can strategize therapies we have and develop new ones. So I'm delighted to call on stage the two moderators. Daniela Sia is an assistant professor and researcher at the Icahn School of Medicine, Mount Sinai in New York, and Annie Babiker is a medical oncologist at the Mayo Clinic. And I will leave the stage to you now. Thank you.
I'll introduce you.
Okay.
So we're going to get a next session of therapeutics, looking at drug development and combinations of chemotherapy to fight cholangiocarcinoma. And it brings me great pleasure to introduce Dr. Nilo Azad, who's a professor of oncology at Sidney Kimmel Comprehensive Cancer Center, where she serves as the co-director of the Development of Therapeutics Clinical Research Program and the Cancer Genetic and Epigenetic Core Research Program.
Thank you very much, and I want to thank the organizers for inviting me here to speak at my favorite conference every year. I've been tasked with spending the next 20 minutes talking about the next generation of combination therapies for patients with cholangiocarcinoma. And I think the last couple of days has made clear the complexity of this disease and how challenging it's going to be to come up with a single combinatorial approach. And so what I'm hoping to do today is to really talk through some of the emerging data that's come and how we as clinical trialists can look forward in terms of what we can do next. So these are my research disclosures and my consulting disclosures. So what's our reality right now in taking care of patients with biliary tract cancer? Well, clearly there have been significant advancements in the last 5 years. You've all seen the number of drugs that have been approved. We have a new first-line therapy after over a decade that involves immunotherapy. And concurrently, because of the increase in understanding and in technology, we really understand the biology of this cancer better than we ever have before, though there is still significant work to be done in trying to dissect the complexity of this cancer as well. The other interesting thing about our cancer is that among the GI cancers, biliary tract cancer has more targetable mutations, more patients that have something that we have a drug for or could have a drug for than any other GI cancer. And so with that truth is still the truth that we are not where we need to be, and survival for our patients is not nearly good enough. What we need is new drugs that hit new targets. We need drugs that are better, and we need to combine them together better. So I wanted to start with a big part of what we've been talking about over the last couple of days, and that's the genetic landscape of patients with biliary tract cancer and how we can use what we know about particular targetable mutations to make combinations that make sense, that have science behind them and rationale behind them. And so in this figure right here, I've tried to highlight what's in red are mutations or abnormalities that we presently have either FDA-approved or guideline-approved therapy for. But none of these therapies are curative. And so combination approaches might be ways to target patients, but better than we have been with the drugs that we have now. And then in yellow, I also highlighted a few other mutations where there are drugs in the clinic or even approved, but for other tumor types, and we haven't yet figured out how to use them for patients with biliary tract cancer. And I'm going to start with IDH1 because there's been some beautiful work about how we might be able to target IDH1 better in the last year. So this is the Clarity trial. Others have presented it, so I'll go through it very quickly. But this was a clinical trial in the second line in advanced metastatic cancer patients who had an IDH1 mutation and were treated with ivosidenib, an IDH1 inhibitor, versus placebo. And this is the updated survival. Now, this trial had a crossover design, so patients who were in the placebo group, once their cancer grew, were able to cross over and get ivosidenib. So it's difficult to show an overall survival benefit when both groups of patients are going to get exposure to the same drug. But when they corrected for that, there was still an improvement in overall survival with the correction. But you can see at the right side of the curves, we still don't get there in terms of curing patients, getting them living years rather than an increased number of months. So Nabil Bardisi's group published last year a beautiful paper that gives us a hint about a new strategy that we might want to use for patients with IDH1. And Nabil's work really looked at multiple components of how IDH1 inhibitors are working using mouse models. But one of the most important things that came out of that paper was that we actually needed an immune system for IDH1 inhibition to work. And what this figure right here is showing, if you look at the two curves that, does this have a pointer? If you look at the two curves at the very bottom, those are the curves that separation is what happens if there's an intact immune system and you give a mouse, or you could extrapolate a patient, an IDH1 inhibitor. That red curve shows how much tumor growth is controlled. But over on the left, what you see is the curves don't separate when a mouse that doesn't have an immune system is treated with an IDH1 inhibitor. So you need an immune system for an IDH1 inhibitor to work. Does this make sense? Is that what an IDH1 inhibitor is? Is it an immunomodulatory drug? Well, what is IDH1? So IDH1, if this is bringing back any troubled memories for people from medical school or other times, it's actually an important protein that's involved in glycolysis, that's involved in how we utilize glucose oxidative using oxygen. And so in the Krebs cycle, IDH1 is an important protein that results in an alpha-ketoglutarate, which is eventually a substrate for energy for a cell. In the setting of a mutated IDH1, what happens is you end up having a buildup of an abnormal protein called 2-hydroxyglutarate, or 2-HG. And what 2-HG does, it affects multiple cellular functions. It infects multiple proteins. It affects epigenetic machinery that changes the way that a cell interacts, and it absolutely affects the immunologic fingerprint of a cell as well. And so in the same paper, Dr. Bardisi's group then took a look at the different cell populations that were affected when an IDH1 inhibitor was utilized. And this is called a UMAP, where you can map the different cell populations and what happens to them when you treat with a particular drug. So what you can see on the right is the brightness in different circles increases when IDH1 is given to these mice. And on figure C and D, what you see is an effector T cell population, a T cell population that's really aimed at killing the tumor, increases with IDH1 inhibition. So that's why an immune system is so important for an IDH1 inhibitor to work. Well, what about our patients with FGFR2 fusions? What do FGFR2 fusions is fibroblast growth factor, so it's a growth factor. It signals through the MAP kinase signaling pathway, and there are many different ways that FGF can be upregulated or increased in cancer cells. The ones that are today's FGFR inhibitors that are approved target are if you have an FGFR2 fusion. So these drugs are now FDA approved. They have been transformative for our FGF2 inhibitor patients. So infigratinib, pemigatinib, futibatinib, the latter two are the ones that are presently, I think, the highest in circulation, showing response rates in that 30 to 40% mark and survivals of patients that are living up to 2 years. But people aren't cured with these drugs. And so these slides are courtesy of Dr. Goyle, who's worked significantly in this area of FGFR2 resistance. And what her team showed back when she was at Massachusetts General Hospital was that patients that are treated with FGFR2 inhibitors develop mutations. They can develop mutations within FGFR2, and these can be gatekeeper mutations, which then don't allow the drug to work as well, to bind and to work as well in inhibiting FGFR2. And what they also were the first to show, this is in cell line models, was that if you took cell lines that were becoming resistant after being treated with an FGFR2 inhibitor, one of the first generation ones, but then treated them with a covalent FGFR2 inhibitor that could bind regardless of the gatekeeper mutation, that now you could get tumor control or cell growth control. And in fact, when the futibatinib data was initially released, they really did show that there were a subset of patients that if they had prior FGFR2 exposure, they still benefited, though the drug does not have indication for that setting. Now, there's a new drug that is presently in clinical trial that's extraordinarily exciting, that's been reported. And this does remake the landscape because the response rates to this agent are over 80% in FGFR2 naive or exposed patients. So if you haven't had an FGFR2 inhibitor before. And I say it remakes the landscape mostly because people are still not cured with this drug. And so we still need strategies for how we can overcome FGFR2 resistance even as our drugs get better. And so this is looking at those response rates. So now we've got response rates of 88% at the phase 2 dose compared to response rates that were a little under 40% of the other agents. So there have been a couple of interesting papers that have looked at FGFR2 resistance and what other pathways are upregulated that we might then think about combining in combination strategies. And so this is Samik Rao-Chowdhury's work, and what his group showed, and this was work done in cell lines, that if you have an FGFR2 fusion positive cell line, that you have an upregulation, an increase in proteins in the PI3 kinase AKT mTOR pathway. And this is even more upregulated as the cells become resistant to an FGFR inhibitor, and that's what you see on the right, where that network increases in terms of the number of proteins that are upregulated in that setting. And so then the next step, which was logical in their work, was to look in cell lines. If you combined mTOR inhibitors, which were the pathways that were upregulated, if you combined inhibitors of mTOR, could you help with this resistance problem? And so the way that these assays are done are that the cell lines are treated with either drug alone or both drugs, and then they look at how many cells die, and they look at and see, hey, is this what we would expect? Is it additive? Is it just the two drugs, 1 + 1 = 2? Or is it synergistic, 1 + 1 = 5? And so anything on the figures on the right that are in the red or in the yellow or orange, that's synergistic. And you can see that when you combine mTOR inhibitors in these cell lines with FGFR inhibitors, that you have synergy the majority of the time, and that's demonstrated here numerically. Numbers less than 1 indicate synergy. So the next rational step for this, we can test it in animals. That's just a cell line. We should test it in a more complex model system and then consider testing it in patients. And that would be a good next step for the work that Dr. Rao-Chowdhury has reported. Now, Dr. Bardisi has also been interested in looking at FGFR signaling and what happens when cells or animals are treated with FGFR inhibitors. His work also showed an upregulation in the same MAP kinase AKT signaling, and also another receptor, growth factor receptor, EGFR. And so what you see on the left is a heat map showing that negative regulators of those pathways, so it's a double negative, negative regulators of these pathways were downregulated with treatment, and so then you had increased MAP kinase signaling, increased EGFR, that's what you see on the right. And when you added an EGFR inhibitor, in particular the drug afatinib, they were able to do away with that phenomenon. And then they were able to show this in mouse models as well. And so this is a mouse model system of an FGFR2 fusion. These two different models that they used, and they showed that when you use the combination of afatinib with an FGFR2 inhibitor, that you get the red lines at the bottom, which is that really beautiful tumor control with no tumor growth whatsoever. So that next step for this would be to test it in patients. Now this is a little bit more complicated because we've got drugs that work, so you've got to have an endpoint. If you're gonna give this to a patient who's never seen an FGFR2 inhibitor before, now you're gonna be aiming for the stars, right? You've got a relay drug that has a response rate in the 80s, so it's very complicated to try to build on something that's so good. On the other hand, if you do this in patients that are already resistant to an FGFR2 inhibitor, then you can test it in fewer patients, and you can, your goal and your benchmark is a little bit lower, and it's a little bit more feasible to do. So I'm very intrigued because a trial is planned. This was also this is a second email to Nabil 5 minutes later. But that trial design hasn't been released yet. So we'll be very interested to see how they decided to target this combination. But this is a new option in clinical trials for FGFR2 patients that's going to be exciting in the next year. And I wanted to touch on KRAS inhibitors too, because this is such an exciting area. About 5 to 7% of patients with cholangiocarcinoma, especially extrahepatic cholangiocarcinoma, have mutations in KRAS. And for 40 years, we haven't been able to target KRAS, but in the last few years, we now actually have a drug that's FDA approved for a particular KRAS mutation that's actually very rare in cholangio and GI cancers. But we now have drugs that are going to be hitting the more common mutations in our tumor type as well. And so I wanted to show this data. This is a drug called adagrasib. Adagrasib hits the KRAS G12D mutation, and in the clinical trial that they reported, they did enroll multiple biliary tract cancer patients. Those are the patients in blue, and you can see these are the waterfall plots showing beautiful responses in patients with biliary tract cancer that have this mutation in this early trial of this drug, and they are going to continue to move this forward as well. Combination therapies are going to be the next logical step for these targeted agents as well. KRAS is a very important signaling protein for cell survival, and it has many downstream and upstream partners that we could potentially target as we go forward. And I expect that we'll be seeing more papers and more work in the next year or two that are really going to explore what would be the most ideal combinations. And our very own Ben Stanger, who is now the chair of this Scientific Medical Advisory Board for the cholangiocarcinoma, published a beautiful paper just a month ago that we needed to mention as well, using this G12D inhibitor as well. And what his work showed was that just similarly to what Dr. Bardisi had showed, these KRAS inhibitors do have an impact on the immune system as well. And so these are figures from his paper showing that immune cells that we want, which are CD8 positive cells that are anti-tumor cells, go up when you treat with a G12D inhibitor, and the cells that we don't want, which are tumor-associated macrophages or MDSCs, these are immunosuppressive cells, they go down, or they change their polarity so that we have the ones that we want more, which are the M1 versus M2 ratios also change favorably. So these have an immune impact that we might really be able to harness as well. He further demonstrated this in animal models where he was able to show that if you knock out an animal's immune system, you deplete these immune cells, you still have a benefit from the KRAS inhibitor, but that benefit is not durable the same way it is if the animal has an intact immune system. And so the next step of this work is going to be clearly to look at how we can combine these drugs with other immune modulating drugs as we think about options for the future as well. So that testing these in animal system. But the other thing that's important for a tumor like ours is that we've got to jump on the train when these drugs are coming out and being tested in other tumor types. This is a very exciting drug in other GI cancers like colon cancer and pancreatic cancer. And so our community has been very vocal in advocating with the company to also test in biliary tract cancer, and so they're going to do that as well. And I think we've got to take advantage of those kinds of opportunities. So I only have a few more minutes, so I just want to talk about other immunomodulatory strategies as well. You guys have seen the TOPAZ data significantly over the last couple of days, so I'll go very quickly that we've got an overall survival benefit. We've got a tail, meaning people at 2 years, there's twice as likely a chance that people are going to live at 2 years compared to patients who didn't get durvalumab. But we still need to build on that too. And so there's been conversation about the combining of VEGF inhibition with immune checkpoint inhibitors. Why should we consider that? Well, there is data that shows that cells that help with presenting antigens, those cells can be matured in the setting of depriving them of VEGF, and that immunosuppressive cells like these MDSCs and regulatory T cells can also be decreased, or the tumor microenvironment becomes less hospitable for them if you don't have VEGF, so if you block VEGF. And so that's why it rationally makes sense to combine these two. There's absolutely been activity in hepatocellular primary liver tumors. And so we have a couple of trials that I just wanted to highlight the data that where there was the LEAP study that gave VEGF inhibition in combination with PD-1 therapy and did show that there is a disease control rate of about 70% in those patients. A separate study looking at a PD-1 inhibitor with an anti-VEGF agent also showed, as you can see in this waterfall plot, multiple responders and significant disease control. Our work at Hopkins has been focused on how we might use MEK inhibitors, which are part of that MAP kinase pathway, in order to modulate the immune system as well. Mark Yarshwin, who is now associate professor at Hopkins, in combination with Greg Lazinski, who is also here, they've really been leading this work, showing first our initial clinical trial 4 years ago now, showing that atezolizumab, a PD-L1 inhibitor, in combination with MEK inhibitors, that those patients were able to go twice as long before their cancer grew. But if you look at the progression-free survival curve, there is a separation, but then it comes right back down together. So we knew we needed to build upon that. And so Lauren Dennison, who was a graduate student in Mark's lab, spent her graduate studies really trying to examine what MEK inhibition was doing and why we weren't getting the responses that we wanted in that initial study. And what she basically was able to demonstrate was that MEK inhibitors actually have a negative immune impact when they're given systemically because of their impact on immune cells. And so we can counter that by giving it with a T-cell agonist. And so in a series of studies in mice, Lauren was able to demonstrate that the addition of a T-cell agonist improved T-cell function as well as survival. And so because of that, we now have an open national clinical trial that's open at many of our area centers that's combining a T-cell agonist as well as a MEK inhibitor, the same MEK inhibitor and PD-L1 inhibitor. And I only have 9 seconds, so I'm going to just quickly touch on vaccination strategies. This is an area that is underexplored in terms of combination strategies, and I really hope that is an area that we'll be able to develop further. At Hopkins, we've been developing a KRAS-specific peptide vaccine. This has been done, work that's been done by Neha Zaidi, and so that has been deployed in pancreatic cancer and in colon cancer, and we are presently excited about the idea of deploying that vaccine along with some additional mutations that are more specific to cholangiocarcinoma, and then running a clinical trial in that as well. So in conclusion, we've had so much progress. It's speeding up. It really feels like we're on an accelerated path right now, but we do need to understand why the drugs that work, why they stop working, and how we can reverse that after patients have had their initial benefit. And what we really need is advocates. We need our physician scientists to advocate within our community about working in this space. We need them to be advocating to pharmaceutical companies about allowing their drugs to be tested in cholangiocarcinoma. By far, I feel like that's the biggest hurdle, is to get access to the drugs. There's so many good drugs out there. And then we need everybody else to be advocating in every way that they can so that we can make sure that this tumor that is not that rare can really get the research that it deserves. So with that, thank you very much.
We have time for 1 or 2 questions from the audience. Please go to the microphones if we have one question. But thank you very much for the presentation. It was very, very nice overview. I guess I can ask you one question while we see if other people have something to ask you. So in one of your slides, if I did not misunderstand, there was IDH plus CTLA-4 based on Dr. Badiz's work, but there was second line with a question mark. Do you mind to argue, like if you see a space for IDH inhibitor plus ICI as first line or second line?
So that, when I was doing my slide deck, I decided to design a trial. And then after that, I reached out to Nilo to ask him if the trial was already ongoing. So I put the question mark there because I don't know what line that they're going to be testing this combination in. But I think that would be a reasonable place to test the second line. Yeah.
Can I ask one quick question? I mean, you mentioned, again, CTLA-4, and a few years back, when we did some transcriptomic analysis in resected biliary tract cancer, we saw that the expression of CTLA-4 in peritumoral area, but no PD-L1, was associated with a poor prognosis and relapse. So do you think CTLA-4 is something that should be limited only to IDH1 tumors, or do you think that with the rise of bispecific products or dual combination might have a role in all other?
Yeah, so that's a great question, and not at all. In fact, I think many of us that work in tumor types that are considered either cold or mostly cold tumors in terms of how active immunotherapy is, many of us are impressed with how well CTLA-4 works compared to PD-1. And for a while, it was thought to be kind of a quirk of animal models, but now I think we're starting to see even more human data where that might be true. Kim Rice-Binder published a really nice paper in pancreatic cancer where she gave PARP inhibitor in combination with CTLA-4 and PD-1, and in fact, the PD-1 PARP combination, those patients did less well than would be expected. Meanwhile, the CTLA-4 combination did much better than would be expected. And so I do think that there is something there, and I'd be really interested to hear Dr. Greten's thoughts on it, too.
I'm going to cheat and ask you a quick question, Dr. Azad. What do you predict the sequence of treatment of FGFR2 fusion is looking? This is almost sounding like, the first, second generation anti-FGFR lung cancer. Do you foresee a future of how we're going to sequence anti-FGFR2 fusions with combinations in the future?
Yeah, I mean, I think that's absolutely an open question. I mean, now with both the relay drug that's already in the clinic and there's some other agents that are coming out that I think we're very excited about too. I think that there's going to be a big question about whether they should come first or whether they should be the salvage regimen after someone uses, one of the more first-generation agents as well. And some of it may also be tolerability and how well tolerated they are as well. But those sequencing questions, I think, can be well investigated, both in clinical trials and then in observational data if it's done tightly as well. But I think that's a big quagmire.
Thank you very much. Thank you for being with us. It is my great pleasure to introduce our next speaker, Dr. Tim Greten, who joins CCR Medical Oncology Branch as the head of the gastrointestinal malignancy section. It was promoted as tenured senior investigator in 2015 and deputy branch chief in 2018. Dr. Greten is co-chair of the Center of Excellence in Immunology and co-director of the NCI CCR Liver Cancer Program. It is our pleasure to have you here. Thank you.
Thank you, Daniela, for the nice introduction. Novel targets for future, what is coming from basic research? I have no conflicts. But we're always looking for patients to enroll. I always have open jobs in the lab. Anybody interested to work with us, always, we're there. And I want to thank Stacy. I think she's just doing a really great job here. So my task is going to be, to tell you what comes from the basic lab. And, this is just, one of the many reviews that we have that shows, how much progress we have made. You just heard this from Nilo, and, we don't talk about chemotherapy only. We have targeted therapy, immunotherapy. So yes, I would agree it's a rapidly moving target, which also tells you they only gave me 20 minutes. It's impossible. There's tons of missing parts. I'm not going to cover everything. And I also don't understand everything. So, I just pick a few things, and either you like it or you don't. So when we talk about basic research, what we really, at least the way I see this, it's a lot of translational research. So it's this interface between doing research where you do cell line work or you would do potentially studies in mice and try to test something, and then you see, if what we find in mice actually works in human. But it's actually more than that. I think the more interesting studies, which are also translational studies, are the ones where you actually go back from the human into the mice. And I will show you some of this today. All right, so let's cut to the chase and, enough of all this introduction. So how do we do this research? And as I said, I'm just going to highlight a few of the studies that came out in the past. One of the advantages is that we have now patient-derived xenografts, and we can make tumoroids or organoids out of these that we can test drugs on. And this is one of those studies that was published in Clinical Cancer Research this year that shows you we get, and this is, I think, an important point also, for patients, that we can actually get the biopsy and can take this biopsy into the laboratory and then can potentially implant this into mice. And then the important and test this. And the important thing is that most of the times these tumors actually really maintain the features of the primary tumor. So if we have our pathologists look at the specimen, they look the same. And if we do the genetic testing, they really resemble what is actually in the patient. So this clearly is a good tool, to study different drugs. And this is one of those evaluations where you either do this, where you generate cell lines and do this in vitro and then test this, or potentially treat mice. And you can even, in this particular system, you can even treat resistant tumors that have been exposed to a first drug. So it's basically like a patient that received a first-line treatment, a second-line treatment. You just heard this, discussion, how do we sequence this and can test this. But what are we targeting? And basically, I'm just going to show you some general approaches that are new. The first one that is being tested, and this is all preclinical work, are fibroblasts. So when you look at the pathologist looks at the tumors, you can see these tumor nests, and you see this fibrotic tissue that is all around the tumor. And there are studies and investigators who actually study this. This is one of them. This is work that comes from the group of Robert Schwabe, where they specifically studied these fibroblasts, and they were able to show that there are different subsets of fibroblasts. And then, again, this is preclinical work, so it's in mice. If you treat these fibroblasts, you can actually, and eliminate them, you actually see that these mice develop fewer tumors. So this is, I would say, a very novel and different approach. You don't even treat, you don't use any drugs that target tumors, but you try to eliminate fibroblasts. The next big point is tumor cell metabolism. And now when I talk about tumor cell metabolism, I specifically don't mean just IDH, but just tumor cell metabolism in general. And this is also something that's being discussed here at the conference. You can target altered glucose metabolism, lipid metabolism, or amino acids. We know that these are general phenomena that are different in tumors versus in healthy tissue. These are phenomena that you find in different types of cancers, but they're also being studied in cholangiocarcinoma. The good thing is that there's actually many, many different drugs and pathways that you can potentially target. You can see on this slide, it's a very nice summary, a review that describes this. All these different drugs are basically targeting different potentially metabolic targets in cholangiocarcinoma cells. So I would say, this is clearly something, to watch out for, and that will bring new treatment options in the future. Molecular targets, you just heard this. Nilo mentioned cholangiocarcinoma is the GI tumor with the highest number of potentially molecular targets. And that really has revolutionized the field in cholangiocarcinoma, I would say. And here I would like to highlight some work that has already been mentioned by Nabil's group, showing that you potentially can make this type of treatment more effective in this case of an FGFR inhibitor by combining it with an EGFR inhibitor, in preclinical models. You can very nicely see this, where he now basically shows the data very similar to the way we actually present clinical data, with waterfall plots, and then obviously here tumor volume, and you can see that this combination is very effective, and then more detailed analysis of the environment helps us understand the mechanism. So I think these are new approaches how we can, in the future treat this. But the most important one is obviously the immunotherapy. And so I would like to spend the rest of my talk to tell you about, aspects of immunotherapy and, how we think this is potentially going to move forward. So when I talk about immunotherapy in cholangiocarcinoma, there are obviously way more approaches than just using one drug or type of drugs that may target the PD-PD-L1 pathway. Currently, there are actually studies available or ongoing where we use now bispecific antibodies to target not only T cells but other cells in the environment. There are other checkpoint inhibitors. There is a lot of interest in targeting different cells in the immune environments, such as macrophages, myeloid-derived suppressor cells, potentially activating NK cells. There's this whole idea of performing adoptive T cell therapy. You heard a lot about combination of PD-1 with other agents or with other approaches. And obviously, there is also the still continuing and rapidly growing field of vaccines. Now, when we do this, I think it's important that we do this similar to what we do when we use targeted therapies, that we potentially strategize and really remember that there's different types of diseases that are all classified as biliary tract cancer, but obviously there's an anatomical classification. There are different molecular subtypes that actually display different immune infiltrates, and then if you just want to go very bland, and I doubt that is going to be helpful enough if you just use PD-L1 expression, but that is obviously another opportunity. The interesting thing is that now with the new technologies where we can really look at these tumors in more detail, we make very interesting observations. So this is one of the studies that I decided to show that came out of Cancer Discovery in last year, where they actually now look at different areas of the tumor. So you can see here, oops, sorry, this was wrong, and now I have to go back, and I don't know, yes. This is where you go in different areas: tumor region, tumor margin, and the non-tumor area. They look at different immune cells using multiplex immunohistochemistry. And then you can really see what we call neighborhoods, so how immune cells with tumor cells and immune cells talk with other immune cells, how this really works together. And here, it's a surprising finding here in this case was actually that there were fewer immune cells in the tumor in FGFR mutated tumor, but this is not the only finding of this particular study. But what I want to tell you is, this is kind of makes us, helps us better understand the specific environment. Now with this, I'm going to move, and I'm just going to show you some of the our own data that we have generated at the NCI, and this is basically, the team that is doing this because I'm just the presenter. We actually started with immunotherapy in 2014 using anti-CTLA-4 in cholangiocarcinoma patients, and this is just an overview of those studies, and then, we decided to combine this with PD-L1 in patients, and then more recently, we did this heroic approach of adding a third drug, anti-VEGF. And I want to show you some of this data because I think it's actually very interesting, and this is very early and very, very preliminary. So initially, we started this in 2014 because at that time, we had a clinical trial where we treated patients with HCC, and we saw responses, like many others later also saw them, and said, well, we should try this in cholangiocarcinoma as well, and it didn't work very well, to be honest. So then we went and said, let's combine it with PD-L1. That was a second study, and again, the treatment was kind of, there were every now and then we saw some responses, but it was not something where I say, I can be very proud of this. So then, in 2019, we started this triplet therapy, and you can see, the treatment regimen that is shown here. And we made the interesting observation that after 7 patients, we saw that all of a sudden, a lot of patients actually responded. This is something that we had not seen before. You see, this is only a very few patients. You see this, 6 patients. 1 patient has an HCC, so you may want to ignore this one. But here we saw in a very small patient population, and this is way too little to make any point that this works. Okay, this is only 6 patients. I just want to make sure that everybody understands this. I'm not claiming anything else. I only treated 6 patients here. And then I make another mistake. I show you a survival curve and compare the survival curve from these 6 patients with what we had done before. Those studies were always about 20, 25 patients. You should not do this. And this is what comes out. Okay, but now I'm getting excited. Okay, because this may be something. But what I didn't show you is, he started this in 2019. Why does he only have 6 patients? The reason is we saw unexpected toxicities. All of a sudden, in these few patients, we saw toxicities that we usually don't find that frequently, especially if we only have 6 patients. We saw patients with myositis, with myasthenia, myocarditis. Some of these patients had all of these adverse events at the same time, and they are dangerous. I don't like it when you have a problem with the heart and the central nervous system. That's not something, you want to deal with. But it shows you that we really triggered something, that we really triggered a very strong immune response, which actually correlates with the outcome. We know this. We know that patients, if they actually develop immune-related adverse events, they are more likely to respond to a treatment. Okay, so this is obviously, something that really caught our interest. And when I say caught our interest, remember my third slide with the mouse? So we went back into the mice now. And this is what I mean by translational research. So we said, is this true or not? It's only 6 patients, and I'm telling you, because I think it's important to mention this is only 6 patients, so don't make anything out of this. But it can potentially help you better understand this. So we went back now into our mouse model, and we did study design, like we have in patients. We used the different models, and I'm not going to bore you with all those details, but we can implant them everywhere into the mouse, either into the liver or subcutaneously. And then, we use similar reagents, anti-VEGF, anti-PD-L1, CTLA-4, the ones that we also use in patients, and try to mimic this. And this is what we're currently doing. Obviously, this is not done, otherwise, it would be published. And the first thing that we saw is there's actually toxicity. So these are now tumor-free mice, and we treat them with this combination treatment. And if we look in lung, we find a lot of immune cell inflammation. If we look in their kidney, we find nephritis. If we look in the liver, we find periportal inflammation. And, and then we have experts who can score this and quantify this, suggesting that it's actually very similar to what we see. And we don't see this with the combinations that we had used before. And then if we go and look at efficacy, again, we can look at this over time. And here it may be easier to see. Indeed, this triplet combination is way more effective than what we see with the doublet. Now, currently, we're studying this and trying to understand, on a more cellular basis, what the immunological mechanism is, why this is changing the immune response so strongly, and I hope that soon I can tell you about this. In the last 2 minutes, I have to thank again the Cholangio Foundation because, I think it's actually very nice if you have one project that you do in mice that Lawrence Diggs did that was supported by the Cholangio Foundation where he showed that, and you heard this before by others, that's why I'm not going to talk about this again, about anti-CD40 as a potential treatment option. And then Dr. Monhead now, she told you about this yesterday, is taking this now into a clinical trial where we hopefully can test if what we have found in the murine system also applies to patients. And with this, I'm going to close and thank my group who are doing all the experiments and all the various collaborators that we have within NIH and outside the NIH. And then last but not least, for those who are working in the field, I just want to shout out that there is a special conference on the cholangiocarcinoma, a FACET meeting, which is going to happen in August. And with this, I'm happy to take any questions.
Thank you. Questions for Dr. Greten? Unfortunately, I cannot see. I might need some help. I don't see. I see Dr. Bardisi. If we can get him a microphone, please. Thank you.
Great talk, Tim. Just a quick question. Do you know the source of VEGF in these tumors? Which cell types are producing the VEGF? Would you happen to know? Either from your single cell work
No, actually, that's a good
models?
point. No, we have not looked at the source of VEGF. What we know is that the VEGF in the serum of the patient is elevated, and it goes, as expected, dramatically down if we treat them with anti-VEGF. I mean, that's not, not surprising. It's just a confirmation. But we don't know what the source is. Others have looked at tumors. I know that tumors can produce a lot of VEGF, but I have not looked in this particular group yet.
Okay, thank you.
Any other question?
I will ask one question, which is a little obvious and probably there is no answer. What about heterogeneity? You introduced the concept of CAF has targets, but we know that not all CAF are good, not all CAF are bad. How do you envision tackling that?
So that's an important question, the heterogeneity. And as you know, I work a lot together with Xin-Wei Wang, who is really the specialist in this and should can answer this probably much better. But what I can tell you is there's also an immune heterogeneity, and what we believe is that you may actually overcome this. So because we know that if you kill one tumor cell, you may actually cause bystander killing, and then a different tumor cell that may be different and will release antigens again, and then you will actually prime an immune response against this. So the nice thing from an immunological perspective, at least, is that heterogeneity does not necessarily have to be a huge problem.
Thank you. Thank you very much.
We're going to switch gears right now from novel combination of therapeutics and immunotherapy targets to actually CARs that are finding their way fast to our clinical trials portfolio within each cancer center, not in leukemia, but in solid tumor. And with that, it brings me great pleasure to present Dr. O'Hare, who's going to talk to us about the potential of CAR T-cell therapy in this disease. Dr. O'Hare is an assistant professor of medicine, Division of Hematology Oncology at the Aberson Cancer Center at the University of Pennsylvania, where he concentrates on the treatment of gastrointestinal malignancies. Dr. O'Hare is a clinical and translational researcher who focuses on immunotherapy in GI cancers. Thank you very much.
Thank you for that kind introduction, and thank you to the organizers for the invitation to speak today about the potential of CAR T-cell therapy in cholangiocarcinoma. I have two tough acts to follow here, so hopefully I can do some justice to what I see as sort of an emerging field in GI cancers in general, but hopefully also in cholangiocarcinoma soon. Here are my disclosures. So overall, I just wanted to give just a brief outline of what I hope to accomplish today, and that sort of is to, first off, give a background to CAR T-cell therapy. As CAR T-cells are not commonly used in cholangiocarcinoma, as we'll soon find out, but they're emerging. I wanted everyone to get on a basic framework of understanding of CAR T-cells, and then kind of using a lot of my experience in using CAR T-cells over the last 10 years in different cancers, especially pancreatic cancer, and in terms of looking at some of the barriers that are set up against solid tumor efficacy of CAR T-cells in solid tumors, including target selection, and looking at the immunosuppressive microenvironment, and then looking at some potential strategies that we're using to overcome some of the CAR T-cell exhaustion that we're seeing within our tumors. And then finalizing on just looking at some of the open clinical trials right now that I hope to continue to expand in cholangiocarcinoma. So looking at what is a CAR T-cell, so looking at CAR, which means chimeric antigen receptor. Basically, it combines an external scFv of an antibody, so something that recognizes a specific target that we want to target in a cancer cell, with the endogenous T-cell effector function. So if you look on the bottom left, you see an endogenous TCR, which is basically the normal T cell receptor that we see in our T cells. And then to the right, how we've modified the T cell receptors. We've taken some of the endogenous components, so for example, the CD3 zeta that's there, and combined it with co-stimulatory domains, 4-1BB mentioned there, but there's also CD28 amongst other ones. And then we've attached it through a hinge region to an SCFV of an antibody targeting a protein that is overly expressed on cancer cells. And so a lot of the work we're going to be talking today is on mesothelin, although there are some other targets that we'll mention there. These constructs are built in the lab, and there's been modifications over the years from sort of first generation, which just had the CD3 zeta, to second generation, which includes one co-stimulatory domain, third generation, fourth generation, and now fifth generations as we're kind of combining more co-stimulatory domains or more chemokine secreting domains within the CAR T-cell construct itself. These constructs are made in the lab, and the way we bring it to patients is that we collect their T cells, as I'll show on the next slide, and we will introduce the construct through a lentiviral vector that's been modified, so it basically will just introduce the construct into the nucleus of the T cell, as shown here. When it introduces it, it's then processed and then able to be presented on the outside of the cell, and now we have a CAR T cell. We grow these up in the lab, and we give them back to the patient. That whole process is shown here in this figure. So we start with a patient who's interested in pursuing a clinical trial. We undergo apheresis to collect their T cells. We select these T cells. We infect them with the lentiviral vector in the lab. We grow them up and expand them. And then once expanded and we do some quality and control, we are ready to give them back to the patient. This whole process takes about 3 to 4 weeks on standard, although there are some ways of actually speeding up this process overall. But that is definitely a pertinent point when we choose the right patients for these trials, because you want someone that is basically able to have their disease controlled prior to enrollment and treatment on the trial. And when ready, we can give them back to the patients, and patients may or may not receive lymphodepleting chemotherapy prior, and you'll see that's variable amongst the trials that we're about to show. And so moving forward and just sort of looking at CAR T cells in general, amongst patients, we know that CAR T cells have revolutionized the management of hematologic malignancies, specifically CD19 CAR T cells, which I did a lot of work with, actually just sort of seeing patients in my fellowship at Penn, as we had some of the first clinical trials there. And now there's 5 different CAR T cell products that are approved for use by the FDA. 4 of them are CD19 based and are approved for ALL, B-cell lymphoma, follicular lymphoma, and mantle cell lymphoma, and one is a BCMA-directed CAR T cell that's approved for use in multiple myeloma. And there's ongoing studies on how to improve the efficacies of each of these CAR T cells in these diseases, but they really revolutionized the management of these patients. However, as we try and translate them to solid tumors, as has been mentioned several times at this conference, there's several barriers that are in place that we need to try and overcome. And that's a lot of the work I've been doing to try and see how, what barriers the cancer itself is putting up against these CAR T cells, and try and see what methods we can use to try and overcome them, and use that as knowledge to hopefully translate these CAR T cells into efficacy for cancers like cholangiocarcinoma. Some of the barriers I'll focus on today is looking at CAR T cell selection, the trafficking of CAR T cells to the solid tumors themselves. You know, one of the easy, quote unquote, easy things about liquid tumors is that when you give CAR T cells into the vein, the CAR T cell sees its target right away because of the circulating hematologic malignancies, but the CAR T cells need to go from the vein to the tumor, and then once it gets to the tumor, it's got an immunosuppressive microenvironment to contend with, as we've heard over and over and over again at this conference. But I think there's lots of strides being made in terms of understanding this microenvironment. Hopefully we can take some of this learning to combine it with CAR T cells to actually make this a more effective therapy for our cancers. So looking specifically at CAR T cell selection, the target selection, we know that targets for these CAR T cells, it's different for some of the other processes like T cell receptor or adoptive cell therapy, but these targets have to be an extracellular membrane-bound protein. And so something that can be recognized on the outside of tumor cells. And the goal is to find a target that is presented on the extracellular membrane of tumor cells, but not expressed at high levels at normal tissue. Because when you give these CAR T cells, it's going to basically see the receptor, see the target, and attack the target. So you have a potential for on-target, off-tumor activity that leads to side effects. We'll talk about that in a second. Looking at the potential targets, looking at the preclinical work that's being done right now in developing CAR T cells specifically for cholangiocarcinoma, I've outlined some potential targets here. And I'm happy that a lot of the same targets that were mentioned in Dr. Greten's talk just prior to mine are also shown here on the slide. But we have one example showing next of EGFR being used. This is something that's sort of universally presented on intrahepatic cholangiocarcinoma, about 50% of extrahepatic cholangiocarcinomas. Although we know that EGFR is also expressed on normal tissues and has the chance for on-target off-tumor activity. A lot of work I've done in the clinic is on mesothelin-expressing tumors and its variable expression, probably more likely expressed on extrahepatic cholangiocarcinomas, up to 2/3 in some cell line assays, and maybe about 1/3 in intrahepatic cholangiocarcinoma. CD133, cancer stem cell antigen, is presented about 70% of cell lines. MUC1, or the mucin-1 gene, also expressed at high levels, and then aberrant glycosylation of MUC1, which I'll show on the next slide, is also expressed about 70% of cell lines for cholangiocarcinoma. HER2, definitely a lower presentation, less than 10% of patients, but mostly in the extrahepatic and gallbladder cancers, and definitely gives a potential targeted therapy from adoptive cell therapy potential there. And then the last but not least, integrin alpha V beta 6 is also expressed at high numbers. And so there's CAR constructs that are designed against all of these targets in the preclinical space, and we'll see that there are some clinical trials exploring these CARs in cholangiocarcinoma or all solid tumors currently. Just as an example of how actually trying to decide which is the best type of target to use, I explore TN-MUC1. So MUC1 is a mucin gene. It's a glycoprotein commonly expressed in normal, healthy, simple, and glandular cells, and a little bit in leukocytes. And in cancers, we find that MUC1 gene is actually aberrantly glycosylated. So we get these TN or STN moieties added on. And so now we have something that MUC1 is expressed on normal tissue, TN-MUC1 is expressed in tumor cells, so it's sort of an ideal target for us to sort of say, okay, this is something that's expressed in cancer cells, not in normal tissue. Maybe we can have some activity with CAR T cells here. And we see here in the lower right a construct that's been made using a SCFV to TN-MUC1 linked by a hinge region to a 4-1BB co-stimulatory domain and a CD3 zeta endodomain. And if you look at the graphs at left, you see that when you use this construct in a xenograft model of TN-MUC1 expressing cholangiocarcinoma, you see increased tumor kill, decreased tumor volume over time, and then at the bottom, looking at the histochemistry, you see an infiltration of the CD3 positive T cells into the cancer tissues of the CAR T cell treated tissues, but not just using a mock T cell without a CAR T cell construct. So proof of principle, and this is moving into the clinic as we speak. The only clinical trial that's actually published right now looking specifically at a CAR T cell in cholangiocarcinoma is looking at an EGFR CAR T cell. I'm using this as sort of an example to kind of point to the fact that we really need to look at the targets closely. And so in this trial, they used the EGFR CAR T cell, and patients received lymphodepleting chemotherapy with nab-paclitaxel and cyclophosphamide, followed by infusion of the CAR T cells, and they were monitored closely. We see a modest clinical activity on the right side. You see one patient with actually a durable complete response, which is quite interesting. 10 patients with stable disease, but this translated into a median progression-free survival of only about 4 months. So definitely some potential movement in terms of making this more efficacious in this population. What I want to point out, and sort of circled on the left-hand side, is the adverse events that we're seeing. And although not all patients develop significant adverse events, we see that there's evidence of on-target off-tumor activity. We know EGFR is expressed on most of our tissues, including the skin, and we see that patients developed over time desquamation, pruritus, gastrointestinal hemorrhage, mucositis, and oral ulcers, some of them significant. And so from that perspective, clear evidence that this target itself may not be the best because there's a potential for significant adverse events. And I'm glad that most of these were not grade 3 or grade 4, but something to be cautious of in the future. In my own experience using mesothelin CAR, I want to point out also the potential that some of these targets that are thought to be just expressed on cancer cells and at low levels of normal tissue, we could still have some on-target off-tumor activity. This is our clinical trial looking at a mesothelin-directed CAR T cell in pancreatic cancer delivered by IV initially in the first cohort. What we saw on trial is that 1 of 3 patients had stable disease when it was given intravenously. Patients did not receive lymphodepleting chemotherapy on this. We did see a good expansion of CAR T cells in the blood, and then as we commonly see, that it falls off about day 14 to day 30. And we see the presence of CAR T cells in the tumor tissue, so we know that these CAR T cells are getting to the cancer through the IV delivery. The thing we weren't expecting was that 2 out of the 3 patients developed significant pneumonitis. I give an example here just on CT scans. We see baseline CT scans at left with patients with pulmonary nodules. That's her site of disease. Day 11, she got admitted to the hospital for some increasing subjective shortness of breath, although no need for oxygen. We got a scan, and the nodules are about the same size. There's a little bit of inflammation showing in between, just sort of throughout the lungs at these 3 representative cuts. And as we progressed, the patient developed worsening shortness of breath, worsening oxygen requirement, eventually developed high-flow nasal cannula, a need for high-flow nasal cannula, and we see that the scans themselves show worsening pneumonitis. And the patient was in a prolonged hospitalization up to almost about a month and a half, and then eventually we were able to wean down the oxygen and get her off oxygen as an outpatient with some improvement in the pneumonitis. This kind of illustrates 2 points. What we know is that mesothelin is expressed at higher levels on cancer cells but lower levels on normal tissue. And we had always thought that this was sort of because of the pleura, the pericardium, the peritoneum. But what we also know is mesothelin is at very low levels in type 2 pneumocytes. And so what we think actually ended up happening here is that when there was some level of inflammation against the mesothelin in these type 2 pneumocytes, inflammation then actually led to more mesothelin production, that then led to more CAR T cell activity, that then led to more worsening pneumonitis. Now, this doesn't happen in every patient, so it doesn't explain everything 100%. And one of the things we were also worried about is by giving this by IV delivery, the first place that this patient, the CAR T cell actually encountered mesothelin was in the lungs, because it went from the IV into the vein to the right side of the heart to the lungs, and then hopefully it was supposed to go back to the left side of the heart and then the rest of the body to the tumor. But the first place it encountered it was in the lungs itself and caused this inflammation. From this, we learned that we might need to actually change our protocol a little bit, so we went to regional delivery, where we deliver CAR T cells directly to hepatic lesions through a specialized device, a hepatic arterial infusion through the TriCellus TriNav device, and then also in the peritoneum using a peritoneal catheter for those with peritoneal carcinomatosis. And what we found is that the CAR T cells obviously made it to the tumor, but part of our big focus in this is actually to look at biopsies here and actually try and figure out what was happening to the CAR T cells within the tumor. So we actually had a little higher levels. It was safer. We didn't see the pneumonitis in any other patient itself, and we're still ongoing in terms of analyzing some of this data. So now, knowing that we can get these CAR T cells to the tumor, now how do we get them to work? And we've heard over and over and over again about this tumor microenvironment, the myeloid-derived suppressor cells, the regulatory T cells, the TAMs, the fibroblasts. All of these forces that are up against the normal immune system are also forces against the CAR T cell. And so we need to think of different ways of actually getting these CAR T cells to be better or combining it with something to make it a little bit more effective. So one logical thought is, can we combine CAR T cells with checkpoint inhibition? And while this hasn't been done in many cancers, it was done at Sloan Kettering using a mesothelin-directed CAR T cell in patients with malignant mesothelioma. These CAR T cells were actually infused directly into the pleura because that's where most patients' disease was. And what we saw is initially patients just received the CAR T cells into the pleura, and then I don't know if you can see the red numbers there, it's a little bit small, but all patients that have the red number actually also received a checkpoint inhibitor. And what you see is that the responses that we see on this were increased in those that received the pembrolizumab, had a median overall survival of about 18 months. And then looking on the lower part of the graph, you see that's sort of looking at CAR T cell levels in circulation. And when patients receive the pembrolizumab, we actually see an increase in CAR T cell presence in circulation. So clearly some potential synergy there. Now, PD-1 inhibition actually does work in mesothelioma, and so further work has to be done here. But I think overall, when we look at our other diseases, such as cholangiocarcinoma, where single-agent PD-1 inhibition isn't necessarily all that effective, we might need to look at some other strategies, and there's definitely several different potential options to look at here. I'm kind of switching gears a little bit to actually, rather than doing combinations with CAR T cells, kind of look what can we do about the CAR T cell itself to try and see if we can actually make some changes to make them more effective and maybe less targeted by this immunosuppressive microenvironment. This is a clinical trial that's been ongoing looking at Gavo-cel, or TC210. It's a mesothelin-expressing TRuC T cell, so it's sort of a modified T cell receptor-based therapy, so not necessarily within the CAR T cell realm, but I thought it was worth highlighting here. So this is basically taking an entire endogenous T cell receptor and fusing to it to target antibodies to target mesothelin. And it works through an HLA-independent manner, unlike some of the TCR therapies that we have in clinic. And this study showed an impressive waterfall plot looking at some responses as well as prolonged stable diseases here in the phase 1 setting. It was also pretty well tolerated overall. I've highlighted on here, despite this trial being open in mesothelioma, ovarian cancer, and cholangiocarcinoma, only one patient with cholangiocarcinoma was enrolled on this trial. And they did demonstrate a partial response to this therapy, albeit it was not significantly prolonged, as they did come off therapy before 4 months because of progressive disease. But this phase 1 trial did lead to orphan drug approval for mesothelin-expressing tumors, including cholangiocarcinoma. They've moved this now into a phase 2 study, and they're using, first started with just a single agent at the recommended phase 2, and now they've actually modified the protocol to now include checkpoint inhibition, looking at nivolumab, as well as nivolumab and ipilimumab in mesothelioma, and then nivolumab in both ovarian cancer and cholangiocarcinoma. And I'm interested to see how nivolumab would work in something like cholangiocarcinoma, where PD-1 inhibition may not be the best option. And it would be interesting to sort of see if ipilimumab may be a better choice in these cold tumors, as I sort of echo what Nilo had said in her talk earlier today. I have 2 more quick points to make. One thing that we also realized, I mentioned in the CAR T-cell trial that we did for pancreatic cancer, one of our big focuses was to see what was going on with the CAR T-cells in the tumor microenvironment. One of the things that was happening concurrent with the trial is in the lab they were studying the effect of CAR T-cells in these mesothelin-expressing tumors. And what they found is that with continuous antigen exposure, the CAR T-cells actually become less proliferative, less cytotoxic, and less effective in terms of secreting cytokines, as shown here. And so knowing that these CAR T-cells, when we give them in humans, see a continuous antigen exposure most commonly, what can we do about potentially finding a signature that we might be able to target? Moreover, actually looking at this continuous antigen exposure, if you actually took away the antigen, then the T-cells actually could rest and could return to actually having high proliferation and cytotoxicity. Looking at these CAR T-cells that had the continuous antigen exposure, we were able to identify CAR T-cell dysfunctional signatures in the lab, and we focused specifically on SOX4 and ID3. 2 transcription factors were upregulated within these continuous antigen exposed tumors. And what we found is if we actually knock out SOX4 and IDA3 or ID3, then that actually led to less exhaustion of these continuous antigen exposed CAR T cells, and so it opens up the possibility that we may be able to translate this into the clinic. To figure this out, though, we wanted to see whether or not these CAR T cells actually had this dysfunctional signature in our patients, and this is a representative slide from one of the patients on the peritoneal infusion of our mesothelin-directed CAR T cell, and we in fact were able to find this dysfunctional signature, including upregulation of SOX4 and ID3 on these CAR T cells. And so current work right now is actually looking at modifying and potentially knocking out SOX4 and ID3 during the manufacturing step of our CAR T cells to potentially be able to bring that into patients to hopefully lead to less T cell exhaustion. And I realize I'm just out of time right now, but I just want to focus on just this one last topic here. It's a new clinical trial that we're opening up for cholangiocarcinoma patients among also with ovarian and mesothelioma, but this is a new sort of novel design looking at mesothelin, a mesothelin CAR. And so what we know is when we take chimeric antigen receptors, I showed you in the beginning, we're basically taking parts of what normally happens within the T cell that are separate functions, and we're combining it all into one construct. And so with this, we wanted to, the work had been done in the lab at Penn showing that actually if you're able to take certain parts of it and keep it separate, we might be able to increase the efficacy of these CAR T cells. And so they designed this thing called a KIR CAR. So basically, they took the killer immunoglobulin-like receptor, which is found in CD4 positive, CD8 positive T cells, as well as NK cells, as well as the DAP12 immunoreceptor. These are sort of separated, and if you actually put them and design them as a CAR T cell and put it into tumor models, xenograft models of mesothelin-expressing tumors, you find that these two components of this CAR combine only when they're actually bound to mesothelin, and then after tumor cell kill, there's no more mesothelin, so they separate. So it kind of recapitulates more of the standard T cell receptor activity. And you see that in this model here of a previous CAR T cell that was a model of mesothelin-expressing tumor that was resistant to the standard CAR T cell, that these cure CARs were able to actually increase function itself. And this has now moved on to a phase 1 clinical trial, which just opened, looking at patients with mesothelin expression. Mesothelin expression cholangiocarcinoma is the part that I'll be running. And this is something that literally we're just starting to screen last week. Patients on this trial will receive lymphodepleting chemotherapy with fludarabine and cyclophosphamide, followed by infusion of these cure CARs, and there's going to be 6 cohorts at a 3+3 design. And so we're excited to see what kind of activity we see in the clinic on these new generation of CAR T cells to try and see if we can increase efficacy. I know I've talked about a lot of barriers to CAR T cells, but there is CAR T cell work being done in cholangiocarcinoma. I think that we're going to continue to learn about them. This is a list looking at my clinicaltrials.org research here of trials that are available. Most, unfortunately, are available in China for those in the US. But in the US, top one is the mesothelin-directed cure CAR that I just mentioned. There's also a couple of HER2-directed CAR T cell trials that are available. And a lot of the other CARs that are mentioned there are things that I mentioned earlier where they had done preclinical work to find efficacy for those receptors. And so in conclusion, we find that in hematologic malignancies, CAR T cells are effective, but in solid tumors, there's a lot of barriers that we need to overcome. And similar to what's been mentioned earlier, we need to find what's happening in other diseases, how we can modify these CAR T cells, what logical combinations can be done within cold tumor microenvironments similar to what's seen in cholangiocarcinoma, so that we can actually make this potentially effective therapy for patients with cholangiocarcinoma in the future. And with that, I'd like to thank you for your time. Thank you again to the organizers for inviting me to the conference to speak, and happy to take any questions. Thank you.
I think we can have time for one or two questions, if that's possible. So I was going to throw out one question. So thank you very much for the great presentation. It sheds light into the future of CAR T cells immunotherapy in solid tumor malignancies. One talk, I think you touched point on it. One talk we always hear about CAR T cells is the CAR T cells exhaustion. And I know in the world of leukemia, they're looking at boosters or treatments to overcome that exhaustion after treatment. Is that something that you foresee will occur in the future in solid tumor?
Booster
Yes.
treatments? Yeah, and so actually built into the pancreatic protocol right now, we actually have the regional delivery with sort of 2 subsequent IV infusions. In the ovarian cancer and mesothelioma trial, they're actually doing 3 IV infusions. And so that's something that we're studying to see if it actually helps with this exhaustion. I think a lot of it's going to be getting that tumor biopsy sort of before you do the repeat, as well as sort of after all the repeats, and sort of see if we're actually having a role on it. We are having potentially some increased clinical activity, although I can't say it's sort of a major change. And so I do think it's going to be more than just doing a booster, personally.
One of the questions is the lymphodepleting chemotherapy, because the issue with the type of therapy is that you have to gain some time because you have to produce the cells, etc. And then the other question is, what type of chemotherapy are you actually proposing to use for lymphodepletion?
Yeah, so those questions I think I could actually spend an entire day talking about, just because it is definitely a source of contention, not only in some of the liquid tumors, but also in solid tumors. What we found in our early clinical trials in pancreatic cancer, ovarian cancer, mesothelioma is actually when we gave lymphodepletion, we actually didn't see much change in either expansion or proliferation of the CAR T cells in circulation, or actually translation to the tumor. So when I designed the pancreas trial, I actually opted not to do the lymphodepleting chemotherapy because there are obviously a potential of side effects that come with that in terms of neutropenia that can happen. The type of lymphodepleting chemotherapy is interesting, and I think that there's variability amongst different centers. There's sort of a new theory that I'm kind of ascribed to and sort of testing out in my pancreas trial right now, is that maybe we actually don't need lymphodepletion, but maybe we need conditioning chemotherapy. And so one of the thought processes I have is actually, can I use the chemotherapy that's actually appropriate for use for the cancer to actually kill off cancer cells, to see if I can condition the tumor or the tumor microenvironment to receive these CAR T cells a little bit better. And so how I modified the pancreas trial right now is actually in the first line. So patients actually get collected before they start first-line chemotherapy, receive 3 to 4 months of chemotherapy, standard chemotherapy for their pancreatic cancer, And then I will work the CAR T-cell infusion at that point after sort of maximal benefit of the chemotherapy to see if maybe I can use the standard chemotherapy as a conditioning regimen to potentially make these CAR T-cells better. And so I don't know the answer to that, but I do think it's definitely something that we are still learning about, and I think we need to still think a little bit more critically, especially as we design trials for solid tumors.
Excellent talk. I have a question, especially regarding improving the efficacy of CAR T. There was a paper that came out of C.D. Chen's lab at Yale last year. I think they looked at kind of a CRISPR knock-in screen, and they found that an enzyme related to protein metabolism, actually when they knocked in kind of like the co-stimulatory molecule that improved CAR T efficacy, effector function. Are you aware of that? And then have you kind of looked into those avenues? And is that type of therapy or modality kind of ready for prime time to be trialed in clinical setting or something like that?
Yeah, so that's a great point, and I think that's where a lot of the work being done in CAR T cells is heading right now. Well, I don't know that one specifically. I know that at Penn we have done CRISPR-Cas9 knockout, not necessarily knock-in, of certain genes. We did a TGF-beta double knockout and actually a modified PSMA CAR in prostate cancer and saw some interesting results there. And so it's definitely something we're exploring, and I do think will be certainly a part of CAR T cells in the future.
I have a quick question. Regarding mesothelin expression in cholangiocarcinoma, one of the challenges we had as a site on the TC210 trial was unexpectedly low rates of positivity. And I'm wondering if there's any data according to anatomic subsite that would help us know which patients to look at.
No, I think that's definitely something that I'm concerned about moving forward with this SynCure CAR trial as well. In the numbers that I kind of showed at the target levels were actually stuff that's based off of cell lineages, sort of preclinical models, or some patient-derived samples, and so it's not necessarily representative of what we see in the actual trials. I had a similar outcome when I was screening patients for pancreatic cancer as well, where I was told 90% by those things, and it ended up being about 40% at the levels we were looking at. And so I don't know what it's going to be. I do think that what we see is that the extrahepatic cholangios tend to have more mesothelin expression than intrahepatic, and so I do think there's going to be an enrichment for the extrahepatics in our trial.
Thank you.
I want to say thank you very much. We appreciate it. Thank you for the great talk.
It is now time for our last presentation from Dr. Sandra Kazak, who unfortunately was not able to join us in person. We will play a video. Dr. Sandra Kazak is a pediatric oncologist and the acting team leader for the gastrointestinal malignancies in the Division of Oncology in the Office of Oncology Products at the Center of Drug Evaluation Research, FDA. Over 15 years at FDA, Dr. Kazak gained expertise in the design and analysis of clinical oncology trials of drugs, biologics, combination therapies, and companion diagnostic for the treatment of cancer. And in the next 20 minutes, she will discuss future thinking for clinical trials in cholangiocarcinoma. Let's play the video, please. Thank you.
Good morning. My name is Sandra Kazak, and I am the clinical team leader for the gastrointestinal malignancies team in the Office of Oncologic Drugs at the Food and Drug Administration. I would like to thank the Cholangiocarcinoma Foundation for inviting me to participate in the 2023 annual conference and for keeping the dialogue with the FDA open. I have no conflicts of interest to disclose. In 2010, the results of the ABC02 trial were published, establishing cisplatin and gemcitabine as the standard of care for biliary tract cancers. It took another 10 years before another treatment was approved for cholangiocarcinoma. Since then, 3 FGFR2 inhibitors, an IDH1 inhibitor, and an immune checkpoint inhibitor have been approved. These therapies represent incremental improvement, but cholangiocarcinoma remains a disease with an unmet need for effective therapies. For more than half a century, the clinical development of anti-cancer drugs followed a paradigm with sequential stages. Phase 1 trials were designed to determine the drug's safety, tolerability, and dose. Phase 2 trials then explored the drug's activity in a variety of cancers, and Phase 3 trials compared the new drug with existing treatments and serve as the basis for regulatory approval. This paradigm requires the conduct of multiple trials and a long time for a drug program to demonstrate that the drug is effective and safe for a particular indication. Multiple trials were required for each disease and multiple trials for each combination. This was an inefficient and burdensome paradigm. Innovative strategies are changing the clinical trial landscape. Advances in our understanding of cancer biology have led to both development of more effective drugs and improved patient selection by early biomarker discovery and development of assays to reliably detect these biomarkers in patients' tumors. The three distinct sequential phases of drug development have therefore become increasingly blurred. Innovative trial designs like master protocols and adaptive trials can accomplish many objectives in a seamless fashion and provide a more flexible and effective way to conduct clinical trials. In addition, through the advancement of biomarker discovery and identification of tumors harboring these genetic aberrations, when the biology supports that a particular genetic aberration drives the tumor growth, irrespective of the tumor origin site, a tissue-agnostic development may be possible. Traditionally, cancer clinical trials have relied on overall survival and measures of tumor growth or reduction to assess the efficacy of a drug. However, benefits are often accompanied by significant symptomatic toxicities. The degree to which a therapy improves disease symptoms and introduces symptomatic toxicity affects how patients function in their daily lives. Patient-reported outcome assessments in cancer trials and evaluation of functions are crucial to truly assess the benefit of any treatment and are being slowly incorporated into trial design. As we are coming out of the COVID-19 pandemic, the clinical trial ecosystem should be changed to address what we learned. Patient-centric, decentralized clinical trials are needed to reduce patient and sponsor burden and increase accrual and retention of a more diverse trial population. The growth in technology, data mining and processing, and other technological advances are opening the doors to incorporate real-world data into clinical trials. For each of these topics, the Oncology Center of Excellence, or OCE, at FDA has already published guidances and numerous papers. There is virtually no area in medicine and care delivery that is not already or will be not soon be touched by artificial intelligence. Analysis and interpretation of information from multiple sources and large databases will revolutionize clinical practice and the clinical trial landscape. None of these innovations will be possible without establishing partnerships between all stakeholders. Although these are exciting developments, any future thinking should be informed by the experiences we have gained on clinical trials. I would like to focus this talk on issues related to FDA's experience gained on multiple oncology trials that should be addressed when considering the design and conduct of such trials. Because of time constraints, this is an incomplete list. These additional considerations include issues related to diversity and representation, dose optimization, development in earlier disease settings, and pragmatic trials, particularly in the context of cholangiocarcinoma. The first consideration is related to diversity and representation. Drug development is mostly a global enterprise. Clinical trials are planned under the assumption that the treatment effect applies to the entire target population, particularly to the regions included in the trial. For a rare disease like cholangiocarcinoma, multi-regional trials are necessary and should be designed to allow for an examination of the applicability of a treatment to diverse populations. The table on the slide summarizes the population by region enrolling the 5 trials supporting approvals for cholangiocarcinoma since 2020, showing a similar enrollment across North America, Europe, and Asia. However, as we can see on the table on the right, although the trials were multi-regional, the population enrolled does not adequately represent racial and ethnic minorities, and results may not be truly informative of the effects of treatments across the population that has the disease. These trials enrolled only 3% of Black patients and only 5% of Hispanic patients. Of note, in the US, the highest incidence of cholangiocarcinoma is observed in Asian Pacific Islanders, a group that was almost not represented in these trials. Furthermore, although the median age of diagnosis of cholangiocarcinoma in the US is between 70 and 72 years old, the median age of patients enrolled in these trials is 59 years, and 61% of patients were younger than 65 years of age. Although data on where these trials are coming from, although data on where these patients are coming from is not available, most of the centers enrolling patients in these trials are not community practices, leaving an important gap in our knowledge of the feasibility of receiving these drugs in the places where most patients will be treated. Diversity in clinical research, including in clinical trials, benefits everybody. Last year, FDA published a guidance with recommendations for sponsors to develop and submit a plan to address inclusion of clinically relevant populations. In December of last year, the Food and Drug Omnibus Reform Act, also known as FDORA, was signed into law, which means that diversity action plans to increase enrollment of subjects from underrepresented populations is now required, expanding the original scope of the guidance. Diversity goes beyond racial and ethnic minorities. Shown in this slide are several OC projects aimed at increasing diversity and representation in clinical trials. These projects have a wide-ranging scope, from working on policy like the implementation of the requirement of enrollment of diverse populations, working on outreach initiatives for patients, advocates, families, and people living in underserved urban and rural communities to increase communication and foster understanding between stakeholders, to provide a framework for concurrent submission and review of oncology products among international partners. In addition, there are numerous publications and ongoing collaborations with other institutions like ASCO, AACR, and others that are aiming to expand diversity. The second area in need is related to dose optimization. Future clinical development programs should be designed to conduct proper dosage optimization and incorporate dose optimization strategies into drug development. With traditional cytotoxic drugs, as we can see on the left graph, dose response and dose toxicity curves were parallel. The default decision was to select the highest dose that was evaluated, reflecting the belief that higher drug doses will have better therapeutic activity. However, as can be seen on the right curve, this paradigm does not apply to targeted drugs. In the table on this slide, we can see that in at least 2 of the approved FGFR2 inhibitors for the treatment of cholangiocarcinoma, most patients require dose modifications, likely indicating the poor tolerability of the studied dose. For infigratinib and futibatinib, as a condition of approval, dose optimization studies to evaluate lower doses have been required. Dose optimization should not be an afterthought. Dose optimization prevents avoidable toxicity, increases uptake of the drug, and improves adherence to treatment. Through Project Optimus, OC is working on multiple efforts to improve dose optimization in clinical trials. In 2023, the FDA guidance on optimization of dosages in oncologic diseases has been published, which includes specific recommendations for each stage of development. Among other recommendations, the totality of the data, including dose response for toxicity and efficacy, and the assessment of low-grade toxicity, which has the potential to substantially impact the quality of life of patients, should be incorporated into dose selection. An important step forward in clinical trials would be a reframing of the timing for enrollment of patients. This slide from the University of Chicago describes the current drug development paradigm in which clinical trials are mostly centered in patients with late-stage disease who have already been treated with one or more prior lines of therapy. However, consideration should be given to design trials that support initial approval in the earlier disease setting, which may provide important advantages. Initiating the clinical investigation of new therapies in an earlier treatment setting can provide more patients the opportunity to receive the investigational drug early enough in the disease course when disease-related factors do not preclude participation in the trial and when the treatment has the potential to alter the course of the disease more effectively. Trials in earlier settings may improve assessment of drug effects. There is greater opportunity to characterize the drug's safety profile while avoiding the confounding effects of disease-related complications or sequelae of prior treatments. In earlier disease settings, drug effects of new therapies can be compared to established standard of care. Randomized controlled trials are less challenging to conduct earlier in the course of advance of metastatic disease due to larger patient numbers and identifiable effective treatments to compare the new drug to, factors which may be lacking in a more advanced setting. And earlier disease setting investigations have the potential to improve general outcomes. With the increase in the number of approved cancer treatments, there is a need to continually optimize treatment strategies for newly diagnosed patients with metastatic disease. While treatments in the frontline setting for many diseases may provide improvement in survival, better treatment options could also potentially replace current non-curative and often very toxic treatments. Project Frontrunner is an OCE initiative to encourage drug sponsors to consider when it may be appropriate to first develop and seek approval of new cancer drugs for advanced or metastatic disease in an earlier clinical setting, rather than the usual approach to develop and seek approval of a new drug for treatment of patients who have received numerous prior lines of therapies or have exhausted available treatment options. The goals of the project are to develop a framework for identifying candidate drugs that are appropriate to initially develop for the treatment of early metastatic disease, taking into account clinical, scientific, regulatory, and operational considerations. Facilitate engagement with drug sponsors and drug development to develop and implement strategies to support approvals in earlier clinical setting, and to engage and collaborate with internal and external stakeholders on related research, policy, and educational initiatives. Although there are innovations in the design of clinical trials in oncology, drug development continues to be expensive and slow. But this approach could be different, and some of these trials can be simplified. Although not generalizable to all clinical trials and mostly applicable to drugs or combinations of drugs, for which there is a breadth of experience and knowledge, a pragmatic approach with limited data collection may be used. An example of a pragmatic trial that shows how a close collaboration of multiple stakeholders, including academia, cooperative groups, NCI, FDA, advocacy groups, and industry working together, is the Pragmatica Lung trial. The Pragmatica Lung trial history started with data from a proof of concept trial, LungMap. LungMap is an NCI trial of precision medicine in lung cancer that tests multiple treatments simultaneously under a master protocol. Patients who enroll in the trial get a genomic profile to determine the mutations of their cancer. Based on those results, patients are matched to a treatment being tested on LungMap and randomized to the targeted therapy, if available, or the control. On the right highlighted, we can see the arm of interest. In this particular arm, S1800A, Patients with non-small cell lung cancer previously treated with immune checkpoint inhibitors and platinum-based chemotherapy with progressive disease were randomly assigned to receive ramucirumab plus pembrolizumab or investigator's choice standard of care therapy. With a goal of 130 eligible patients, the primary objective was to compare overall survival using a long-run test with a one-sided 10% type 1 error. In the 136 eligible patients, the hazard ratio for survival was 0.69, which suggested that there was an improvement in the patients who got pembrolizumab and ramucirumab versus standard of care therapy. In addition, the safety profile favored the combination ramucirumab-pembrolizumab over the standard of care. The results from LungMap S1800A presented a unique opportunity to test run a pragmatic trial. Based on the promising survival data from the exploratory trial, strong safety data collected over years for both antibodies, and the need for effective treatment for these patients, the Pragmatica lung trial was designed to remove many of the barriers that prevent people from joining clinical trials. For example, allows the participation of patients with lower performance status who are more representative of people with advanced lung cancer. The trial designers also cut out many of the extra tests, data collections, and secondary study goals, and only grade 3 or higher adverse events will be collected. Patients will be enrolled across the country, treated, and followed up in the same manner they would be in real-life settings, effectively integrating clinical research in the clinical care environment. Of note, although multiple stakeholders were involved, less than 7 months after discussions on trial design started, the trial opened for enrollment. To conclude, as we embark in a new, exciting era of clinical trials, the successes and failures of our experience should be applied to better serve our patients. I would like to acknowledge my colleagues for their insight, help, and providing slides. And once again, would like to thank you very much for your attention.
With that, we close our session. I would like to thank all our speakers, the chairs, and Stacy for organizing such a beautiful conference. Thank you all.